Hi, so welcome back to another Poultice Pod AS revision lesson. This week we're going to try and tackle some of Miss Double's topics at AS, and we're going to look at topic 6, exchange. So I apologise in advance, this is not my usual unit. Miss Double I'm sure would do a much better job, but I know the basics and I can help you recap some of the things that you've done with her last year. So to start off with, we've got to talk about why organisms need to have exchange surfaces, and this all comes down to the phenomenon of surface area to volume ratio. So if we compare the surface area of a cell or an organism, a whole organism, to its volume, we can actually start to make some predictions of whether it will have an exchange surface or not. Larger bodied organisms tend to have a much lower surface area to volume ratio than smaller bodied organisms, because as you increase the size of the organism or the size of the cell, the volume increases much more rapidly than the surface area does. As a result, that surface area to volume ratio begins to decrease. So for single-celled organisms, for simple organisms like bacteria and single-celled eukaryotes, they don't require any exchange surfaces because they're a single cell, and therefore they have a very large surface area to volume ratio, and they can simply rely on diffusion through their cell surface membrane. There's no other barriers to diffusion, so the rate of diffusion in and out of the cell is very rapid. As a result, they can exchange all the materials they want. They can get oxygen in, they can remove waste like carbon dioxide, no problem, just by purely relying on diffusion through their cell surface membrane. Larger bodied organisms, on the other hand, do have this very low surface area to volume ratio, and as the organism gets bigger, that ratio decreases even further. As a result, they've had to evolve exchange surfaces to maximise the rate of diffusion across them, and basically effectively increasing their surface area internally or externally depending on the surface in question. So typical exchange surfaces in multicellular organisms might be the mammalian lung, fish gills, the respiratory system in insects, leaves, roots, the digestive system, the small intestine in particular, are all examples of exchange surfaces. So what do all exchange surfaces have in common? Well, your GCSE knowledge is usually sufficient here, and maybe a little bit of a top-up. So the first thing to remember is they're all got very large surface areas. This will increase the rate of diffusion, okay? They tend to be very thin, usually 1 cell thick. That gives you a short diffusion distance. Therefore, you're getting a much more rapid rate of diffusion across that exchange surface. Okay? They tend to be located close to a transport system. Now, in animals, that may well be the blood. And what that does is that transport system is continually moving away any substances that are being absorbed or bringing in substances you want to remove. As a result, you always are maintaining a steep concentration gradient between the blood and the exchange surface, like the alveoli. Often we might see a phenomenon called ventilation, which we often associate with mammalian lung breathing in and out, but this also may well apply to the gills, because fish are constantly pumping fresh water over their gills, so effectively they're ventilating them. And again, what this does is it maintains that steep concentration gradient. So there's your overview of why we need exchange surfaces and the adaptations they have. Now we're going to move on to look at some specific examples of how different organisms have evolved exchange surfaces to overcome this problem of surface area to volume ratio. Okay. All right. So let's look at the exchange surface in insects. I'm going to cut to a picture now to give you the basic kind of overall structure of them. Hopefully you're familiar with this. So insects have a series of holes running down the sides of their body. These are called spiracles. They allow gases to move in and out of the insect. Those spiracles are then connected to tracheae, little tubes, and those tracheae branch off into finer tubes called tracheoles. And those tracheoles will actually extend within to the body tissues of the insect, and they will terminate there. And that is where the actual exchange takes place, at the end of those tracheoles. The tracheoles are also typically, when the insect is not being overly active, filled with fluid. So how do these work? Well, instead of actively moving air in and out, which insects can do, but if an insect's at rest or is not overly active, how air moves in and out of the insect actually comes down to respiration in the cells. So as the insect cells are respiring, they're going to be using the oxygen to break down glucose. So the concentration of oxygen inside their cells will decrease. There is going to be a higher concentration in the air outside the insect. So we create this concentration gradient, and therefore the oxygen will move along the tracheoles into the respiring cells, basically creating yourself a concentration gradient. And the opposite will happen with carbon dioxide. The respiring cells are going to produce CO2. As a result, we're going to end up with a high concentration in the cells and a low concentration in the air. So therefore that carbon dioxide will move along the tracheoles and out the spiracles, down its concentration gradient. If insects are requiring more rapid exchange because they're being more active, they can do what's called abdominal pumping. Now this is usually only found in larger bodied insects because, again, as you get a larger insect, the surface area to volume ratio decreases. So they've got to have other mechanisms to overcome that problem, and abdominal pumping is one where they do it. If you've ever seen a wasp on a summer's day and you might see it landing on an object, you might see its abdomen appear to be throbbing. You might think, well, that looks nasty, it wants to sting me. The reason it's throbbing its abdomen is not as an aggressive thing, it's actually abdominal pumping. It's probably flying around all over the place and is having to get more oxygen and more oxygen in and more carbon dioxide out. So what abdominal pumping does, it changes the internal pressure inside the tracheoles. So by pumping the abdomen, we're increasing the internal pressure, and that's actually forcing more carbon dioxide out of the insect's body. Okay? So another adaptation they have to deal with high levels of activity is what they can do is, if they're being overly active, then their cells are likely switching to anaerobic respiration because they're not delivering enough oxygen. And this actually acts in the insect's advantage here, because what it does is lactate gets produced inside those cells, and the lactate will lower the water potential of those respiring cells. So that water I mentioned that's found inside those ends of those tracheoles will actually move into the cells because it's obviously going from that high to low water potential via osmosis. And what that does is, by removing that liquid, it's easier for the gas to move, the oxygen to move through air than it is through water, through a water medium. So therefore we're getting a much more rapid uptake of oxygen, which will hopefully compensate with the insect's higher activity levels. So that gives you a nice little overview of the insect. Let's look at the mammalian lung next, and look at the kind of structures what are going on there. Okay, so in the mammalian lung, I find actually a lot of this is very similar to GCSE and are therefore quite straightforward. So firstly, we want to think about the structure of the lung itself. The lungs contain a variety of different structures in this whole respiratory system. So obviously we've got connected to our throat, to our mouth and our throat, is the trachea, the windpipe, which obviously has these rings of collagen which help support and keep that airway open. As the trachea then extends downwards into the chest, it divides into two to form the bronchi, and those bronchi further subdivide into smaller tubes called bronchioles, and then terminating in those bronchioles are the alveoli. The average human pair of lungs will have up to 300 million alveoli, so we have many of them, which of course by just having many alveoli we're going to greatly increase the surface area for exchange, and of course alveoli is where the exchange takes place. The actual alveoli themselves are folded to increase their surface area further. They're only the actual epithelial cells that make up the lining of the alveoli are 1 cell thick, and they're very thin cells at that, to create a short diffusion distance between that and the blood. They've got elastic fibres in them to enable them to stretch during inspiration, and then also to recoil during expiration, breathing out. Alveoli are surrounded by a very complex capillary network, and that, coupled with the ventilation of the lungs, helps to maintain that steep concentration gradient. So oxygen will continuously diffuse into the lung during inspiration, and carbon dioxide will diffuse out during expiration. Okay, so the next thing you might want to think about is, how is inspiration and expiration achieved in the mammalian lungs? Well, this is done through two sort of groups of muscles. One is the diaphragm and the other are the intercostal muscles. So during inspiration, breathing in, if you feel your own rib cage and you breathe in, Nice deep breath there. You'll feel that your rib cage should be moving up and out. Now, the reason your rib cage is moving up and out is because your intercostal muscles are contracting, and therefore they're pulling the ribs up and outwards. At the same time, my diaphragm will flatten as well. It will contract and it will flatten. Both of those things will increase the volume inside my thorax. If the volume increases inside, the actual pressure decreases compared to atmospheric air. So I create a pressure gradient between outside and inside, and as a result air will actually flow into my lungs. And then the opposite happens during expiration. So those intercostal muscles and diaphragm muscles will relax, the ribs will move down and in, volume decreases, pressure increases, and therefore we've got a pressure gradient going outwards and air is forced out of the lungs in expiration. Okay? So there's a little overview of the mammalian lungs. Let's now look at fish gills and how they're adapted for exchange. Okay, so with gills, we have a series of gill filaments, and on those at right angles are the gill lamellae, which increases the surface area of the gill filaments significantly. Fish have many gill filaments, which again further increases the surface area. The blood that flows through those gill filaments also creates a nice steep concentration gradient between the water and the blood. Okay, and the blood that's always arriving is low in oxygen, and the blood that's leaving is always high in oxygen. So that helps create that steep concentration gradient across the gill lamellae. Fish also have a countercurrent system, which means that the blood and the water are flowing in opposite directions. So as a result, we maintain a steep concentration gradient across the whole gill lamellae. Fish can also use their operculum, these sort of gill flaps, and their mouths to actively pump water over the gills, thus ventilating them, which will also ensure that oxygen-rich water is moving over the gills and the oxygen-poor water is moving away from them. So that's the kind of idea of ventilation again. So there are just a very basic overview of the respiratory systems in fish. Let's look at the exchange surfaces in plants, because I feel this links quite nicely in terms of gas exchange. So leaves are quite extraordinary structures because they've got to be able to carry out photosynthesis, and they also need to be able to exchange gases with the atmosphere. They need to be able to absorb carbon dioxide from the air and also release oxygen as a byproduct of the photosynthesis reaction. So leaves are very thin by design, because what that will do is it will always provide you a very short diffusion distance between the air and those mesophyll cells inside the leaf. To allow carbon dioxide to move into the leaves, we need lots and lots of stomata, again creating a shorter diffusion distance, because generally speaking we need to be as close to the air as possible. So but there is a downside with having stomata, because water vapour will leave the plant. The rest of the leaf itself is waterproof because of the waxy cuticle. So on one hand we need stomata to allow carbon dioxide to diffuse in, but too many stomata will cause too much water vapour to evaporate out of the leaf. So what plants often do is they need to be able to open and close their stomata to control and prevent too much water loss. Now leaves have what they use is potassium ions and their guard cells to regulate the size and shape of those guard cells, which will cause the stomata to open and close. If I want my stomata open and therefore allow CO2 to diffuse in, what the plant cell will do is it will actively transport potassium ions into those guard cells, which will lower its water potential. So water will move into the guard cells from other cells nearby and cause them to become turgid and effectively open up that stomata. If the plant is losing too much water, then we can actively transport those potassium ions out of the guard cells, which will cause the water potential inside to increase. Therefore, water will move out of the guard cells, causing them to become flaccid, and that will actually close up the stomata. Plants have other adaptations to deal with dry conditions, and plants that grow in very dry conditions we call xerophytes. So typically what plants are trying to achieve here is about reducing their surface area and about creating a smaller gradient between the inside and the outside of the leaf to reduce water loss. So what you might find with a lot of plants that are growing in dry conditions is they might actually have leaves with a very low surface area to volume ratio, and might have big fleshy leaves as well, so they can store that water. By having a lower surface area to volume ratio, they're less evaporation of water. In terms of lowering the concentration gradient of water vapour between the inside and the outside of the leaf, there's many different ways that the plants can do this. Some have rolled or curled leaves, and as the water evaporates out of the stomata, it fills up that little interior space. It creates kind of a little microclimate, if you will, which with a high concentration of water vapour. Now, as the water vapour inside is kind of trapped, it will actually lower that concentration gradient between the inside and the outside. So less water will actually evaporate. Plants also can do this with having hairs or sunken stomata in pits. And again, the same thing is there. It traps that humid, moist air, and it lowers that concentration gradient between the inside and the outside of the leaf, therefore preventing the plant losing too much water. So we've looked at gas exchange systems in a variety of organisms, but we also need to consider exchange in other aspects. So let's move on now to have a look at the small intestine. Okay, so the small intestines. First of all, we'll talk about the structure of the small intestines and how they aid in the actual absorption of our digestion products, and then we'll look at actually how the different key sort of macronutrients are digested to aid in their exchange and absorption into the bloodstream. So like before, the small intestines are an exchange surface, so they've got to be able to have adaptations like the other ones to resolve this problem of surface area to volume ratio. So the first thing you'll notice is that the small intestines themselves are folded into structures called villi. So these little finger-like projections all the way around the sides of the small intestine walls and the ileum. The cells that make them up, those epithelial cells that make up the lining of the ileum, the actual membranes are further folded into microvilli. So this increases the surface area significantly, massive surface area, something like 75 square metres, if I remember off the top of my head. Inside each villi, inside villus I should say, there's a capillary network, which again provides a good blood supply, which is always going to maintain a steep concentration gradient between the inside of the lumen of the intestines and the blood. So therefore that will aid in rapid diffusion. The epithelial cells that line the villi are one cell thick, again providing a short diffusion distance. Another issue that can come about with digestion is that the blood actually might begin to become get equilibrium with the lumen. So in other words, the glucose, the concentration of glucose in the lumen might become equal to that of the blood. As a result, we haven't got that steep concentration gradient anymore, and we're at equilibrium, so diffusion will no longer take place. To overcome this problem, the epithelial cells are well adapted for active transport, so they're going to contain many mitochondria, which is going to provide a source of ATP for active transport to move those glucose molecules against their concentration gradient, which links to the exchange topic that I teach you with co-transport, so you should know all about that already. So let's move on to now the process of actual digestion, what it means in the enzymes and how they work. Okay, so digestion is, in basic terms, the breakdown of large insoluble molecules into small soluble ones that can be absorbed into the blood. When you consume food, you have to do both chemical and physical digestion to break down that food into small enough molecules that are soluble that can then be absorbed. Mechanical digestion is any kind of physical breakdown process like chewing, any kind of muscle contractions involved in the digestive system like peristalsis and things. And then the chemical digestion is through the process of enzymes and other associated compounds like acids and bile. They necessarily don't directly break down the acid and the bile, don't directly break down the chemicals, but they can aid in the function of our enzymes. So the three main macronutrients you need to be aware of in digestion are the carbohydrates, proteins and lipids. So carbohydrates are broken down by carbohydrases. The most common one that you should be familiar with is amylase, which is found in your saliva. It's also produced in the pancreas, and the ileum cells in the small intestine can also produce amylase as well. What amylase does, it breaks down starch into maltose. This is where a lot of students go wrong. because at GCSE we make it a bit easy for you and we say amylase breaks down starch into glucose. But in fact, that starch to glucose is actually a two-stage process requiring two different enzymes. So amylase, remember, breaks down starch into maltose. Maltose is then hydrolyzed by maltase into two molecules of glucose, which can then are small and soluble enough that can be absorbed into the cells by facilitated diffusion or co-transport, depending on the concentration gradient. So that's pretty easy, the carbohydrates one. Protein digestion is a little bit more complicated. Now, protein digestion will actually begin in your stomach, and you have various proteases there that their optimum pH is pH 2, which is acidic. The acidity can actually help aid the breakdown of the proteins because it will start to denature them at those extreme low pH, will denature and start to break hydrogen bonds within the protein, which will make it easier for the actions of enzymes. So there's three families, if you like, of proteases. We've got our endopeptidases, our exopeptidases, and our dipeptidases. Okay. So what the exopeptidases do, they actually start to break down the protein molecules at the ends. They're snipping off sections at the ends. The endopeptidases start to break it down in the middle of the protein chain. So they're effectively breaking it up into smaller and smaller chunks. Dipeptidases, what they will do is they will break down small sections, basically two amino acids into one. Okay, so now we've got small individual amino acids. These can then be absorbed through facilitated diffusion or co-transport, again depending on the concentration gradient. The one that's probably the most difficult to get your head around is the digestion of the lipids. And the actual digestion bit itself is relatively simple, the chemical bit. It's more the absorption that I find students get a little bit muddled with. So your average lipid, like a triglyceride, is made up of glycerol and 3 fatty acids. And lipases can break those ester bonds to give us glycerol and fatty acids. And another substance which I mentioned earlier, bile, will aid in this process. One, it neutralizes the stomach acid, thus creating the optimum pH for lipase, but also it's an emulsifier. So it helps to stop the lipids forming one big blob. It increases their surface area by turning them lots of little droplets, stopping them joining together. The bile salts will also bind to the fatty acids and glycerol, forming these little droplets called micelles, which then can actually fuse with the epithelial cells in the small intestine, and then therefore they release the fatty acids and glycerols into those epithelial cells. Then they will travel into the endoplasmic reticulum, which will package up those fatty acids and glycerol into little vesicles that we call chylomicrons. Those chylomicrons will then be transported out of the cell by exocytosis, so using ATP from respiration, not into the blood, but actually into the lymphatic system. So a different kind of absorption method there compared to the others. So that's it really for the exchange, as a brief kind of overview to kind of refreshing your memory on some of the bits you can't remember. So hopefully that was useful to you guys. So this is Dr. P signing off. Have a good one. Cheers.